Thank you for joining our webinar today on discovering the Atlas of Living Australia. My name's Christy Spire and I'm currently the team leader of Nature Stewardship at Green Adelaide. And it's a pleasure to host the 5th instalment of our seminar series for 2026, which we run to help share the great work that happens right across Adelaide to look at how we can make it cooler, greener, wilder and more climate resilient. And as many of you know and work with, we work really closely with the First Nations of this place, the Kaurna people, and we love acknowledging their connection to country and the way that they have so much to teach us all. Ngadlu Tampanti, Ngadlu Kaurna, Yatanga Imparenti, which in Kaurna means it's good that we acknowledge that we're meeting on Kaurna country today. And also acknowledging that their language has been revitalised over the last season as well, and it's awesome to have the privilege of learning it alongside many Kaurna across Adelaide today as well. So I hope that you can acknowledge and find out more about where you're meeting from today and which country you're connected with. This session is being recorded and will be available on the Green Adelaide YouTube channel, and we've got awesome episodes up there from over the past 3 years now that you can explore. But let's get into today's session. And my slides have stuck, so we'll jump right in. So whether you're involved in conservation, environmental management, community greening projects, citizen science, or simply have a passion for understanding what nature is around you, we're confident you're going to really value today's talk on the Atlas of Living Australia, often referred to as the ALA. The ALA is an incredible resource which brings together millions of biodiversity records from researchers, government agencies, museums, community groups and citizen scientists across the country. So today's webinar is really designed to help you make the most of the resource, and we're very fortunate to be joined by Kylie Morrow and Juliet Seers from the Atlas of Living Australia, who will guide us all through the platform and share their expertise. They will show us how to find species records in your local area, identify threatened species, navigate the different tools and access data that can support your planning, reporting, conservation projects, community initiatives, or just your general interest. So thank you for being here again. Kylie and Juliet will talk for about 40 minutes and then we'll have time at the end for questions. So none of you have microphones, but you do have your chat boxes. So you are welcome to add any queries, chats, links, etc. in that chat box as we move along the session, and then I'll come back at the end to facilitate a question time with Juliet and Kylie. Let me stop sharing and I'll allow you to start sharing.
Thank you so much for having us. It seems like we have a really good turnout. I can see there's about 50 people online now. And yeah, today we're going to do a bit of a mixture of presenting about the ALA and demonstrations. We're hoping everyone gets to walk away having learnt something new or with some food for thought there. I'll also echo that beautiful acknowledgement of country that Christy did. I'm joining from Naarm or Melbourne, the land of the Wurundjeri Woiwurrung people of the Kulin Nation, and would also like to pay my respects to them and elders past and present. My name is Juliet. I work for the ALA as the Training and Outreach Coordinator. I also co-lead our industry engagement and our data mobilisation program, which helps people get data into the ALA, and our Indigenous Partnerships program. My background is in biodiversity and sustainability. I actually started off working in waste education after I finished university. Then I went overseas and spent some time doing conservation education and snake rescue and working as a safari guide in India for a little while. And then I came back and got this cool job that I love, and I get to talk to people all the time about the ALA and how they can use it and get the most out of it. And it just makes me very happy. So I'm glad to be here. And I'll pass over to Kylie to introduce yourself.
Thanks, Juliet. And yeah, I also would just acknowledge that I'm coming to you today from the lands of the Ngunnawal and the Ngambri people, so here in Canberra for me. And so I'm Kylie. I'm the ALA's digital product specialist. What that kind of means, I do a bit of everything, but I would say that I sit within our systems team at the ALA. We maintain a lot of the tools and the services, some of it kind of behind the scenes as well, that keeps the ALA running. Like Juliet, I also have a pretty varied background, although mine's a little different to hers. I have both kind of that design, sort of coming in through graphic design, but then also a bit more broadly, and then also that biology background and doing a bit of conservation genetics as well. So my role at the ALA kind of blends together that tech focus with that goal of delivering data to help achieve better biodiversity outcomes. So that's something I'm really passionate about. And I'm also really passionate about just inclusion and usability as well, to make all of this easier for our users now and definitely very much into the future. That's something that I'm working towards. So I'll hand it over to Juliet to get us started.
No worries at all. So we thought we'd start by there's some people joining who I assume know a lot about the ALA and some people joining who might not know anything about the ALA. So I think we've got something here for everyone, but I wanted to make sure everyone was on the same page to start with. So we've got a little bit of an intro. The ALA is a website that allows you to access biodiversity records from researchers, government, ranger and community groups and citizen scientists, sorry, I should say, natural history collections and herbaria in one central location. It's free to use, and our users are quite broad. Our primary audience is researchers and decision makers, like those who work in government or inform decision making through things like environmental consulting. But community groups and land and sea managers and citizen scientists are also really important stakeholders for us, and they do overlap with research and decision making in various ways too. You can use the ALA to look for where a species occurs or what species occurs in an area, and we'll do some demonstrations later to help show you how to do this. On the ALA, you can currently access over 181 million records about living organisms across Australia, including our external territories. I wouldn't recommend looking at them all at once though, because it may slow down your computer a little bit, but we'll show you how to narrow it down so you can get to a more manageable amount. When I talk about records, what I'm talking about are occurrence records. So this is point data on a map. So on the image on the right on this slide, that's an example of point data, those orange dots. Each point gives an indication of where a species has occurred. In this case, it's pygmy moss in South Australia. So each of those orange dots is a record of where pygmy moss has been observed or collected. Usually the minimum requirements of a record are that it has taxonomic information like species or genus, a location and the date. Some records may have more information than this, and in some instances, particularly with historical specimens, some may have less information than this. There may be times, for example, if you look at a map on the ALA, you might notice a species record in the middle of Australia that you know isn't meant to be there. That occurs usually with quite historical museum specimens that might not have had specific coordinates associated with them other than being labelled Australia, particularly if the specimen was taken by an overseas institution. It's something just to be aware of when you're using the ALA, because sometimes that might look quite odd, especially if it's something from the sea and you're seeing it in the middle of Australia. But now that you know, it just helps you. We just like to be transparent about some of those quirks in the ALA. Those records are still useful for some people, so that's why we still allow them to be so accessible on the ALA. And in terms of the type of data that these records come from, we have specimen data. So that comes from living or preserved specimens or material samples, and they're from partnerships with museums and natural history associations. So we have them both locally in Australia. We have really good relationships with our natural history collections groups and herbaria, and also overseas. And then there's also observation data. So this is from things like surveys or citizen science. It's where a person has observed something with their eyes, but not collected it necessarily. And that type of data can be associated with things like sounds and images as well, to verify the record is what it says it was at the time. So you can also use that type of information from the ALA too, which is helpful if you're putting together educational materials. Sometimes you might need images of a species, and the ALA can be a really good spot to go for that. And so these 181 million records we have on the ALA come from over 800 data sets. And that means that without the ALA, to access all of the same information would mean knowing all of those relevant data sets and tracking down who to contact to obtain access to that data yourself. So how does all of that information get into the ALA? We have data that comes in lots of different forms. So the specimens, observations, and then we have more dynamic emerging data types like camera traps, acoustics, and eDNA as well. These feed through to what we refer to as our data partners. So they're the column in the middle. You might recognize some of those logos and institutions, and not listed here, but also one of these data providers is South Australia's State Atlas, BDVSA. And then those groups feed through to the ALA. So this is kind of a high level overview of what we would call a data pipeline, I suppose. And some data is classified as sensitive. So each state and territory has a sensitive species list. These lists contain species that have been identified as being sensitive due to things like limited remaining habitat, easily disturbed habitat, poaching risk, or high susceptibility for things like biological contamination, if there's a particular disease presence that people are concerned about. Sensitive species have their coordinates obfuscated or generalized, which just means that the dot on the map is less precise, and that's in order to protect that species. So in South Australia, sensitive species are obfuscated or generalized to 10 kilometres. An example of a sensitive species is the hooded plover, which is in the image here. I love hooded plovers, so I just saw an excuse to use them and thought they'd be a great inclusion, as they are gorgeous, but they are quite sensitive because of how their nests are quite open and exposed on the beaches. So when you see a point of a sensitive species like a hooded plover on the map, you can think of it as being a bit fuzzy, I guess. So that makes it more difficult for someone to be able to directly pinpoint where that species is. And that's in order to give it a level of protection. The radius of the species, so that dot on the map, will be increased, and the point displayed on the map then will be positioned somewhere within that new radius, but not at the actual direct coordinate. If you're a researcher though, or a government user, or someone with a genuine need for the exact coordinates, you can request access for that through our restricted access species data service, which I've got the website listed on this slide here. That's an agreement we have. That service is organised by the ALA along with all the state government institutions, and that's where we kind of manage all those requests to make sure the people who do need those accurate records can get access to them. I'm now going to jump into a demonstration. Have I missed anything at this stage, Kylie? Seems okay.
No,
No?
I think that's going well.
Good. Okay.
I think you've covered everything off.
Woohoo. Okay, I will share screen again. So this is the ALA. I'm going to be looking over here because that's my second screen. Sorry. This is the ALA's homepage. You go to ala.org.au. I'll start by going through how to search by species. I had an example of a species to look at. Oh yes, I wanted to look at the Cape Barren goose, because I discovered that during my South Australia research for this presentation. So you can type in a common name for a species or scientific name. If you're not sure of the scientific name, Google's a really good friend for finding that out. And then you just click search in this bar here. It'll then come up with this here, this page. So this is where it's now searched all of the ALA for what could that search term possibly refer to. And so the top result here is the one we're after, the Cape Barren goose. And if I click on that there, it comes up with what's called the species page. On the species page, you can get images and you can select them and learn more about them if you wanted to use them. I think almost all of them are Creative Commons, if not all of them. You can see the licensing associated with the image if you click through and click view details. And then, yeah, you can use them for educational materials and stuff. You'll also see where it's available compiled distribution maps. So this one is by BirdLife International. And then you can see the points on the map there. So these are all the records we have of the Cape Barren goose on the ALA, including one of those points in the middle of Australia, like I mentioned. There is a possibility that that's a real point. Geese can migrate and maybe they've stopped over there, but it's worth clicking on it if you were interested just to confirm if that's the case or not. You can also get other information where it's available, so taxonomy and history. In this case, it's got some Indigenous name information and descriptions. If you then wanted to view and download those records, you click this button here on the right, and it will load this page, which has all of the records. You might want to do some further filtering before you download. So perhaps you're thinking, okay, I'm interested in the Cape Barren goose, but I don't want all of these records. I just want records from my local area. Oops, sorry. You can click on draw a polygon. And then once you click on that, you're able to create an outline of an area of interest to you. So I'm just kind of making this up for the example, but when you reselect that first point, it will close the polygon and it shows you how many records. So we've got 1,643 records of the goose here, and I can click search for records in this area, and now it's going to narrow it down. So now the only records visible are those ones in that area of interest to me. I could do further filtering if I wanted. If I was like, I'm actually only interested in records from January or from a particular date range, or I'm only interested in records with images or from particular data providers, you can do that, or you can also do the reverse. So if you click choose more, you can exclude certain items. So if you potentially like, I'm actually not interested in records from these providers, you can do that as well. And from there, once you've got everything how you want it and you've got the data that you want to export, you can click download. You'll need an account to do this. It's free to create. And from there, you can download them as occurrence records. Either of these formats you can use. I say the full Darwin core is quite detailed. It depends on your use case. For me and for most users I encountered, the legacy format can be a bit more intuitive with the language that it uses and the column names and things, but you can play around and try both and see what just resonates with you a bit more. Both of them, if you export them as a CSV, if you're someone who uses other platforms like QGIS or ArcGIS to do mapping, you can then incorporate them as a delimited text layer into maps that you might create there. Or you can just have the records to do whatever else you would like to do with them. It's up to you. When you go to download, you select a reason. There's quite a few there. Do select the one that makes the most sense for you, because it does inform us and help us understand our users and what types of changes will be most useful for different people when we make changes to the ALA. And then you click next and it'll run the download. You can move away from the page and it'll send you an email once that download is ready for you. It will also send you what's called a DOI, or a digital object identifier. That's a unique link. That means, for example, if you used that data in a report to inform some findings, you could put that link in the report, and that allows someone else to be able to look at the query in the ALA you use to inform those findings. So then if they want to validate what you've said, or they want to understand better how you came to that conclusion, it allows them to replicate that search themselves, or it allows you to do that again. Say if you've got annual reporting or something you need to do and you want to repeat the same thing. So that's one method of searching. So that's by species. Another thing we thought might be of interest to people is to search for what species occur in an area. So we have this tab, and back on the homepage at ala.org.au, we have this tab for government and land managers. If you click on that and then click this one, which is Explore Regions, it comes up with a tool that gives you different options for ways to search for different regions. So you could do all of South Australia, or if you were interested in a particular local government area, or Commonwealth government area, or by a geographic region, you can search that way. So I'm going to do by a geographic region and select the Air York block. You can also zoom in as well, and you could select by scrolling this area on the left, but you can select on the map as well. So the Air York block. And so this now shows me all the records in that particular area on the ALA. So it's already done that area filter for me. And from there, you can do some further filtering. So if you're like, I'm only interested in mammals, or I'm only interested in flowering plants, you can do that, or I'm only interested in insects. So for this example, I'll just look at insects in the area. And from here, now that I've narrowed it down, I'm going to click View Records. If you were still just interested in everything, you can keep it at all species, and that's no problem. But I'll select insects and then View Records. And it comes up with that same page we had before. So if you remember, there was the list of all the records for the goose before, but this time it's got all the species in that area. And you've got the same filters on the side, so you can apply different filters according to what's most useful for you. You can look on the map. You can look at images as well. So it has lots of images from specimens, and it'll also have images from non-specimens, so from observations. And from there, you can click download. In this instance, you can do both. You could do the occurrence records, the same as in the last example, but you can also do a species checklist. So what that will do is, instead of downloading every individual record, instead it will provide you with a list of all the species in that area or in that query you've put together, and it will have counts next to it. So how many records associated with that species were in that search. That can be useful for some people, particularly community groups and consultants can find it useful when just trying to get an initial understanding of what could be in an area before doing a survey. And it's just that starting point to kind of help you understand what to look out for. Doesn't mean that's all that's in that area by any means. And the same thing, you'd select the reason and then select next, and it will go through and it produces that DOI for you once more. We do have how-to videos on all of these too. I know I've kind of gone through them all quite quickly, and Kylie's about to do another example too, but we will pass those on to Christy to share afterwards. But anyway, I will pass over to Kylie. So that's that's the end of my demonstration.
Thanks, Juliet. I'll just get my screen sharing up while I get started. Okay, excellent. We should all be seeing the ALA homepage there. I'll just get my screen rearranged so I can see a few faces, or just a few people there. Excellent. So yeah, that's been a really good overview of what Juliet's presented there as ways of searching the ALA if you're looking for a particular species or a particular taxa, so going in with a name. And then the other way there, if you want to go more by a region or an area, there's a few other tools and ways to do that, but they're two really great entry points. What I wanted to cover today is another way that kind of combines those two approaches. It kind of gives you both as well, and that's using ALA Lens. So I'll intro it a little bit and then I'll show you how to navigate to it. Essentially, it's an interactive map-based tool. You can explore ALA biodiversity data, look at it in context, and with full credit, it was developed by Mitchell Whitelaw and Adrian McKenzie. They're two professors at the ANU, so the Australian National University in Canberra, and that was for our ALA team, and it was completed late 2024. And it was around this idea of having a generous interface, so kind of a usability concept, but basically having something that's really, really rich to explore and discover, and you can click around and just keep finding more great things while you're exploring, which you can do in the ALA, but the lens is perhaps a bit more experimental versus the ALA is more focused around going in with a specific query in mind. So like we've just seen. So the way that you can navigate to it, and again, I can make sure that these links are shared in the follow-up email and other resources, but you can go from the ALA top navigation menu. So you can just go into resources and then ALA labs to begin with. And so that's actually our science team. They have a lot of great blog posts, especially if you use our Galar package. If you use R or Python, that's a really great place to check out. But that's a different topic. That's an aside. You can scroll down from there and go to ALA Lens. And then that gives us our Lens landing page. There's a bit of a description there, gives you a bit of a preview of what you might see. And then there is a mention of some data stories, which I'll come back to a little bit later at the end, just quickly. But what we're looking at mainly is this Lens interface. And so there's an option here to use my location if you want to do that. If you kind of just want to explore and you want a bit of inspiration, a bit of inspo, you can just load up the page. You can just refresh it and see where you end up, because it will choose from one of a set number of kind of pre-selected locations there. So you can see that there's a map there, and you can kind of just click and drag to pan around. You can zoom in, you can zoom out. We've actually landed in South Australia, which is pretty cool, because I was testing this out a few times earlier today, and I was ending up all over and then navigating back. So I'll come back and we'll do a bit of a search, but just to kind of scroll through and show you what we've got here. We've got latest occurrences for a search area, so they're individual records. We've got two different tabs here. One is Discover Species, one being Explore Data. And then while we're looking at the Species tab, we've got various filters here for different life forms with, great little icons there as well to make it really friendly. If you don't want to go in by name necessarily, we've got bubbles there showing different species that come up. And then if someone did want to go in by search, we have a search bar as well. And then just to touch on that data tab, which I'll also revisit, you may want to explore potentially by filtering by life form, data resource. So we've got SA fauna, SA flora there coming up, or by conservation listing based on the national listing. So let's just do a query. So we'll come back to our species tab, and we want to look, in a bigger area here. So I'll center it roughly around Adelaide and hit search. And then that's a pretty big area. So you can just click and drag it in and out to change your search radius. I'm going to make it, something like that. And I'm just going to zoom in a little bit so you can see what we're looking at. And And so the first thing you'll probably notice is this sort of honeycomb hex map, and that's just around the density of the number of occurrence records. So like with a lot of maps, probably pretty intuitive in some ways, that it's the darker sections are where there are more records concentrated, and then the lighter sections where there tend to be fewer records. And then each of these little map markers here represent a species occurrence record. And if I zoom in a little bit more, just depending on everyone's screen sizes, we've got plants, for example, showing up with a little plant icon, and you can click through and that'll go through to the ALA record. We've got fungi here. Looks like we might have a shark or a fish. We've got birds and so on. And then we can also look through these records here if we wanted to maybe zoom in and have a look at the picture, see who submitted it. If it's come via citizen science, for example, it'll be their username and then the date and so on. So yeah, very much sort of those occurrence records that you see in the ALA more traditionally. But let's say, okay, well, we want to do a bit of exploring. So maybe, I don't know, I love reptiles. I studied them. So I'm going to go digging into some reptile data. So we're still looking in this search radius that we had above. And what we'll see if we filter by reptiles is that our filter is going to pop up here at the top, so we can always keep track of that. We can see our common species here, you know, things like various geckos, bobtail, and so on. So they're just most frequently observed. So when you kind of look at the ALA and you've got a search radius, that's the most common species that you get records for. We've got distinctive species, which I'll come back to, but then we've also got our threatened species there. So these are our species that are featured in national lists, so like EPBC. You can click through if you want to learn more. You can mouse over them just to sort of get a sense of what's there, or you can click on one and then that will apply the filter at the top if you want to look at that on the map. With the distinctive species, though, I think that's really, really cool. And I think it's one of the really unique things about the lens. And Mitchell in particular, I think he's done a really great job of this, in that it's meant to show what's observed more often in this area. So I guess if we were to explain it a little bit more scientifically, it's occurring there more often than it is across the rest of Australia. So it's kind of characteristic of that place, or you might even say local frequency is highest compared to their global frequency or frequency across Australia. And so again, in this example, pygmy blue tongue, as I would have probably expected, it's going to be pretty distinctive in this area because I wouldn't expect to see it around the rest of Australia. And so if I then zoom up, scroll up and then zoom out a little, we can see that we've got our records there. North of Adelaide, we've got our density of where those records are, pretty well centered, and then we can go through and we can find particular ones. We can click through to the ALA if we want, and then when that loads up, that's an example of an occurrence record there. So that's come through for that species. We can see it's come in factually conservation as well, and we can see it's come in via SA fauna, so through the department there. So if I just remove those filters, and then I'll just quickly pop across to the data tab. And something to keep in mind with Lens is that when you go between the two tabs, it can reset your filters. So it's not necessarily keeping them on when you switch between. And maybe now we just want to look at data that's coming into the ALA, and it's in this same region. And so we can see that there's quite a lot of birds, which is pretty characteristic of the atlas. So maybe let's have a look at, we can look at our reptiles again, let's say. So we've filtered it down, we've got our reptile filter on, and again, we've got that area up there. And we can see that the top data resource coming in there is actually SA fauna, which is probably not surprising too. And then we've got others like South Australian Museum, iNat Australia as well, iNaturalist. And then if we wanted to filter by conservation status, we can. So we can look at our critically endangered. We've got, we've actually got some earless dragons coming up as well, if we were to scroll through. So it can be really, really cool to explore and just kind of see the shape of the data that's coming through in the ALA. So having looked at that, I might just quickly pop across to our data stories. So if we come back up, we've got 3 here. There is also a link at the top there if you want to go in that way. But one that's really interesting is this digging into data piece. And so it's a really great way of, it's just loading there. Yep, there we go. We can sort of see the various data providers and what's coming in. So we can see, for example, eBird Australia is a very large data provider to the ALA with the most observations. You can scroll through, or you may want to filter a bit. So museum potentially as an example there. And then you can also look at data resources over time. So in this one, I'll clear that off. So again, we can see eBirds coming up there because it's quite a big one. But what's interesting is maybe we look at something like herbarium. We can see if we're looking at these records over time, going from sort of past into now and into the future, we've got sort of those 2000s and a little bit before in the 80s, we've got our herbarium records where things are, perhaps collected, being digitized, whereas we might look at something like more of a citizen science one. Yeah, so iNaturalist Australia is here. And we can see that those records are really concentrated more, down that pointy end of more recent history as well. So it's just something really interesting. I probably won't go too much further into it because I think part of the beauty of this is just exploring it yourself. But there's, some great records here spatially that you can look at with gang cockatoos. And there's these great observations here where keywords have come through when users have described what they've seen. So maybe they were in a tree hollow, 2 of them together, maybe the type of tree and so on. So lots of really great things there. But I guess that's a really good segue talking about the shape of data in the ALA, that I'll hand it back to Juliet and I'll stop sharing.
We're just on the final end of our presentation, and then we'll jump into questions. So do pop questions in the Q&A box, which is next to the chat, if you have any. We just wanted to touch on data bias, or these are, you can think of them as data gaps. It's something to be aware of in the ALA, or really any large data set will have bias of different kinds, but particularly it shows in some unique ways when it comes to biodiversity data. It's just something to be aware of when you use it. Kylie mentioned with Lens, like it's not surprising often to see quite a lot of bird records. And so I'll talk about why that is a little bit here. So this data visualization here was created by my colleague Sandia. So that she's done that in an effort to communicate how some of these bias kind of appear in the ALA. We have a lot of richness and uniqueness in Australian species, with estimations of about 85% of all Australian plants being endemic. But you can see here there's quite a glaring gap when it comes to representation of this uniqueness in national data sets like the ALA. So starting with overrepresentation, almost 60% of the 181 million records in the ALA are of birds. Magpies alone, for example, make up 1.5% of all records on the atlas. They're a very loved species throughout Australia, which is very nice, but there are a lot of records of them. In terms of representation in records counts, so that first graph I had is now on the left, and I've got a new one on the right. So counts of records on average per species. The number of records per species on average is over 100,000 for birds. But when you look at things like invertebrates, so insects, it's fewer than 200 records on average per species. What we record tends to be correlated with perceived attractiveness. You also see this, often people talk about this when it comes to funding and conservation. Birds and mammals have higher numbers of average records per species than things like invertebrates, plants, and fungi. And even among vertebrates, you find things like marsupials are recorded more often than things like rodents and bats. So for groups with good coverage, like a lot of bird species, we can model distributions and track changes in abundance over time. So when I looked at that goose before in the species search, there was that compiled distribution map, and we're able to do that when we have enough data. But for a lot of other groups, like invertebrates, we have much sparser information. So what that means is you're not able to draw such concrete conclusions about where they are or where they might be. If you take insects as an example of an underrepresented group, the amount of information we have can vary enormously by order in this case. So here we've got each of these large squares made up of smaller squares represents an order within insects. The dark green squares show the percentage of species in that order with more than 10 records for that order of species. The lighter green squares show the percentage with at least 1 record. So for pretty and easy to spot records like dragonflies, which are the ones circled on the top top left, we have at least some data for most species. You can see that there's a lot of green and a lot of dark green. But when you're looking at the total number of records for insects in the when looking at the total number of records for insects in the ALA, dragonflies make up 45% of all insect records in the ALA. And if you compare that with beetles, so that's Coleoptera, which is the one circled on the right, they are the most species-rich insect group with over 25,000 species in Australia, but we know almost nothing about over 2/3 of them in ALA data. They make up 18.8% of all insect records in the ALA, but we only have more than 10 records for 27% of these, compared to 88% for dragonflies. So a flow-on impact of this means that entire chunks of the insect world can be essentially invisible in national biodiversity analysis, and even more so in things like conservation decisions. Secondly, there's geographic bias. So on this slide, you can see a visualization focused on our most recorded group, which is birds, to help illustrate this. To create it, we started by creating a species list for each region, drawing on data in the ALA to produce an anticipated list of species that are likely to be in that region. Each hexagon was assigned a percentage reflecting how many of those species have actually been recorded there in records on the ALA. It provides a simplistic snapshot indication of where there is a good amount of data and where the gaps are. You'll notice that these gaps tend to align with things like logistical constraints. So remote areas are harder to reach, so there are fewer records because there's fewer people living there or passing through or able to access them. And conversely, some areas are better sampled because of things like compliance and development-driven surveys being concentrated there, or simply being in an urban area where there's just lots of people to be able to log those records. And many less charismatic or poorly sampled taxa and the regions they inhabit are even more underrepresented, which leaves significant biodiversity gaps if we rely solely on the available data for things like conservation planning. So we had the bird data visualization that's now on the left of this slide. And bird data, as I mentioned, makes up about 60% of the ALA's records. But you can still see they have quite visible geographic gaps, particularly around the Pilbara, the Simpson Dune Fields, and the Great Victorian Desert. And when you produce the same map but for reptiles, these geographic data gaps are even more stark. So that's now the map on the right is for reptiles, with significant regions having 0% completeness. About 2 cells, I think, on the entire map indicate, 100% completeness. It's not a perfect model, so there's definitely some more details there, but just showing better coverage. So these are just some examples of spatial bias that we wanted people to be aware of when using the ALA. And when you use things like lens, you'll see the hexagons all had different colours. The deeper colours represent more dense clusters of records, I believe. So you'll see that around urban areas a bit more. The reason we raise it is just to emphasize that the ALA is a tool you can use, and it's really fantastic, and many find it very helpful, but it is a starting point, and a lack of records for a species in an area doesn't mean it isn't there. We just might not necessarily have data for it yet. So that's the end of our presentation, and we're ready to jump into Q&A. I just had a few links here, and I'll pass them on to be sent out after the presentation. But yeah, if you're interested in providing data in the ALA, it's something we're happy to support you with. We have different ways of doing that. We also run training for institutions or community groups if it's helpful for them and you have a more specific use case, and we have a general support email as well. That's all.
Fantastic. Thank you so much. Yes, unfortunately, we can't see all the lovely faces online. It's a bit disappointing with this kind of webinar setup, but we assume that you've all got lovely faces. We just can't see them. But yeah, thank you so much for that. Yeah, awesome summary and all the details, and I hope everyone is inspired to explore afterwards. I certainly am. So yeah, we do have a few questions in the chat and also in the Q&A. There's one person, Dizzy Dyer, said that there's a new one, catalogue.com.au for caterpillars, that I believe that's been launched here in South Australia. So is that a data set that is connected to the ALA? Are you aware of that one?
Not at the moment, but
Not yet.
will.
Yes.
I can look into it if it's brand
Yes.
new.
Yes, it's brand new. Yes, I think that there was a local
great.
group here that just we're launching it. Potentially this month or recently as well. So yeah,
Awesome.
though if anyone's online that's been involved with that process, it'd be great to hear more about it. And I think you've jumped in and answered some of people's queries already, just around the generalization points, but I'm not sure if there's something that might want to be explained to the general audience in terms of the size of the points and what that refers to.
Sure. So the for the sensitive data question, so the point is generalized, which means the radius, I guess I explained, becomes bigger, and then what happens is the point that you see displayed on the map on the ALA is randomly assigned within that new given radius, so the radius will no longer directly direct you to where that species is. So yes, it does what Peter was asking about, which is
referencing.
Good.
Okay. Awesome. No worries. And from Craig, are you able to explain why sometimes the user finds multiple name types for same species when using biocollectors and science platforms?
Yeah, I can have a go. BioCollect is a bespoke product on the ALA. It depends on the project you're working within, I would think, but I have asked our team. I think some people might be offline, so they haven't responded yet, but I do know in that
Awesome.
Taxonomy is always changing. We do have a great ability on the species pages where you can navigate names, both historical names for something and the current accepted name. And what might be happening on the back end there is, in order to be more it has to kind of consider what other people might call it. And if there's a recent older name, it might still be bringing that up just because people might not yet be familiar with the new name. That might not be the reason, but feel free to contact that support@ala.org.au email and we can get to the bottom of it. I did email the BioCollect team just before to try and get an answer. I thought that I might get one by the time we got to the question, but I haven't heard just
no worries. We know who Craig is. We can follow him up, and I'm sure he'll follow up through there as well. So yeah, please do that. Do that, Craig. Oh, awesome. Doreen's worked out her question. Are you able to filter by date of record or date range? I just worked it out. Thank you. So you must have answered her question along the way. There's a question here just around someone who kind of gave up on ALA when my iNaturalist records didn't appear in ALA and vice versa. So just a question around that, understanding that are they shared across, or are they supposed to be shared?
They are. So there's on iNaturalist, there's different licensing options you can choose, and they're separate for both images and sound, like media that you share. So on iNaturalist, you have to share some media, like a photo or a sound recording at least, to be able to have the record reach research grade and be verified as what it is. When you do that, there are specific licensing conditions associated with the record itself and the media that you've associated with it. So you can set them separately. I know this because my husband's a big iNaturalist user and a wildlife photographer. So what he had happen is he had everything set to not Creative Commons because his photos are protected for his work. But what the implication had been was that then his records weren't being shared with the ALA. You can change the licensing separately so that your records can flow through to the ALA. So it might not currently be set as a Creative Commons license, and that can be changed in the settings in iNaturalist, and it can explain to you the different implications of choosing different licensing. So that's kind of the answer to that.
Right. And I think you've just answered Andreas's question that following up in the Q&A section around just, because, they've noticed that same type of thing, but I think it was linked to the media and the Creative Comms. So hopefully that gives you enough of a direction to go in. And as Juliet and Kylie have said, please follow up with any specific follow-up questions after this as well if you don't quite know how to do it or can't see how to do it in those two sections as well. And Samantha, I think that goes, for the same as well. For your question, who should we contact if we have queries relating to the use of ALA? I use ALA in the classroom with students a lot, and sometimes we have little problems that we need to solve quickly on the day. Is there a quick turnaround, or do you have a bot yet? No.
Kylie might be better to answer that one.
Happy to jump in there. We don't have a bot yet. We have a fantastic user support team, particularly one person, Tim. He's wonderful. If you're getting something's going wrong, it's not working as expected, a bit of a bug or something like that, definitely do email through to that support email, and we can have the various teams who look after those products, those particular functions, look at it. If it's something that you keep getting regularly, I guess we'd also be really keen to hear about it as well. And maybe there's something that we can look at a bit more proactively, because that would also be really helpful. So either way, do please let us know. But certainly that support email is a really good point of contact too. Right.
Awesome. And Nat's got a few questions here, which I had similar types of ones as well. I think we've gotten through everyone else's, but in terms of, is there an easy way to identify which species have been previously observed in an area that are no longer being observed? And if so, how would you suggest going about that kind of search?
So you could probably, I'm just going to have a quick look here. Okay. What you could do, it's not a perfect method, but if you go into the regions tool that I was showing before, it does have this date range slider. What you could do is do view records, and then you could do like download it as a species list, like I said before, and then you could come back to it and remove kind of recent, like depending on what you're considering to be no longer appearing in the area. You could adjust the filter and see how that species list changes, and then I guess you would compare the two lists to see where's that missing element there to get an idea of what hasn't been recorded for a while.
I just add a bit to that as well. Something you could do in regions. So if you were to perhaps, like Juliet, if you're on insects there at the moment, or if you wanted to pick, say, one of the groups within plants, and then that time scale along the top, you can actually hit the play button, and then that will start to play through the various time periods, and then you'll see the records appearing on the map. So depending on how focused you want to get with it, that can also be a helpful tool to just visualize it a little bit. And you can obviously hit pause you can hit stop to restart as well. And you might want to then, pause it and grab some of those records too.
True.
Yeah, great. It's a good one. Brilliant. And then a little one around the beetles, and I suppose this is what you're talking about in terms of, data sets and the complexities around all of these things. But is the lack of data on beetles likely to be more a result of their location, possibly away from citizen scientists, or because people are less interested in them for whatever reason? I assume both, but interested in what you and the team there think is playing the bigger impact.
I think it's a multifaceted thing, definitely. There's an element of one thing. I know we did this for National Science Week a few years ago. We did a session for citizen scientists on taking good photos, particularly of insects for iNaturalist. So something I learnt there was it's really important to take a photo of the rear end of a beetle. Apparently, it's all about that, helps you identify. So take all angles. I guess what I'm getting at there is if you are uploading things through citizen science, a good way to ensure you're getting to species level, which helps us fill those gaps and know where they are, is to be getting as many photos as possible so an expert can come on and go, this is what that is. Another thing could be a lack of interest. They're smaller. Not everyone likes beetles. I love beetles, but they're not for everyone.
Yes.
So I think location can play an element, but there's also a lot of things just in very accessible areas, and you'll see a news story that's like bug that's never been recorded in the wild suddenly seen in urban areas, just because it hasn't been logged before. So
So good to learn how to make sure that if you are uploading, that your uploads are successful to help with fulfilling that kind of bigger picture as well.
Yeah, definitely.
Yeah. And I'll just jump back up into the other comments just to make sure I haven't missed any other ones, but we're almost at time. I think we've answered that one. Nat did have a question around how long does it usually take for an iNat record to appear on ALA? Is it actually verified before it becomes live? Is there just to explain that process?
It's about a week usually, and it does come through both at research grade and before it reaches research grade. It used to be just research grade observations. Now what will be shown to you when you first do the search on the ALA are research grade ones, and non-research grade things are kind of filtered out. You can turn it on though, and the reason we started doing that is because there are some species groups that there might be very, very few, maybe one person in the world, like particularly with insects, like one person who actually is the only person who could accurately identify if it is
those
thing. And so with biosecurity and things like that, we determined it was better to allow those records for niche things come through so the people with expertise can still access them than to not have them at all, kind of.
And is there a process
clearly marked?
Yes. And is there a process in terms of, because this is open source data as well, are those experts verified by yourselves, or how do you become a verification expert?
iNaturalist has a verification community. So
that's right.
Yeah. So there's a few rules around it. It's mostly a lot of either really passionate researchers or retired taxonomists sitting on there and going through and doing a really good thing for research and for enthusiastic people identifying things. It has to also rules around, so it can't just be one person says, yep, this is it, and then it goes ahead. It has to have two people agree on what that identification is. And then if a third person jumps in, or a fourth person, and says, actually, no, it's not, if there's dispute, it has rules as well around, then it might bring it back to genus level or something like that, because it'll go, there's some contested information here, I'm not sure. So it's still quite cautious as well.
Yes. Yeah, great. But that occurs through the iNaturalist
That's
platform
correct.
rather than that all happens before it goes onto the ALA. Yeah,
correct.
Right.
Yes.
Yeah. Great to hear about that process. And you may have mentioned at the beginning, but how long has the platform been around and evolving, and what was the impetus for it to begin with?
So the ALA has been around for, I did have a slide with a timeline and I ended up taking it out.
Sorry, I'm just interested.
I think it's, no. I think 12 or 13 years, Kylie.
Okay.
Yeah, roughly 2010, maybe a little before 2010, but really informally. We typically say around then, because we did have our 10-year anniversary about 2020. It's been a really interesting journey. I don't know, Juliet, did you want to jump in on anything there?
You can talk to it. But I was just going to say we probably started off mostly catering to herbaria and museums and stuff was our primary audience. Yeah, absolutely. They were a really big driver behind it, and they're still very important data providers and partners with us today. So we definitely work with them, but then we've obviously expanded a lot more into things like citizen science and so on, and really just cater to such a wide user base. So it's really great
it's huge.
Chat with you all today.
So it's been quite a steep climb in terms of all the different partners and people that you work with. So awesome. Well, we are at 5, so thank you so much to Kylie and Juliet for your time and your experience and sharing that with everybody, and to have this as a recorded resource going forward is fantastic for the Adelaide and South Australian community. And we'll make sure that we follow up with an email with all of the awesome links and other things so that you can jump into those. And if you did want any of those more practical sessions to dig in a little bit further, do contact the team at ALA. And our next webinar we're very excited to announce is going to be part of Adelaide's first Climate Action Week, which is happening in the last week of July and the first weekend of August. And this time we are going to jump in with planning for biodiversity, to cool cities and strengthen food systems, with one of our awesome local lecturers and researchers, Dr. Douglas Bardsley from the University of Adelaide. So please join us for that one on the evening of Wednesday, the 29th of July. But we will make sure that we send out a link to that one and some more information as we know your calendars get full. Thank you so much again for joining us today, and have a safe journey home, or if you're already at home, snuggle up because it looks pretty dark and wild out here in Adelaide. So thank you so much, everybody. See you next time.